My name is Lorin Golden, not to be confused with one of our speakers today. I'm one of the co-chairs of the MIA, the Models, Inference, and Algorithms seminar series here at the Broad in the Eric and Wendy Schmidt Center. And it's my pleasure to, I guess, kick off a new semester of MIA talks. I'm really excited here to start a new semester here, a new academic year. And I think it's going to be a really good time. We have a lot of interesting talks lined up this semester, and we're going to be continuing on into the spring as well. We have our first set of speakers here, and I'm going to have Sebastiano introduce them as our host today.
Hi, good morning. I'm Sebastiano, postdoctoral fellow here at the Eric and Wendy Schmidt Center. So welcome if you've not been to the MIA talks before. There are two talks today, one from 9 to 10 by Davide and another one from 10 to 11 by Lorin. It's an interactive meeting, so if you feel like you have a question, raise your hand. I'm going to come around and give you the microphone. It's important you ask the question in the microphone for people online. And yeah, I think that's it. So essentially always the seminar runs on Wednesday. Next week, actually, it's an exception where the seminar will be run on Friday in the Broad Auditorium in the first floor. Yeah, and so without further ado, I think we can let Davide speak. So Davide is a grad student at the Polytechnic of Turin in Italy. He conducted a research stay at MIT last year for 6 months, where he started collaborating with Lorin and with the ex vivo group more generally. Lorin will say a few words about that later. And he is interested in deep learning, both theoretical and more applied. Today, he's going to cover more the applied side of it. And the title of his talk is up there, Infrastructure and Modeling Challenges in Single-Cell Omic. The floor is yours.
Thank you. Thank you very much for the introduction. So thank you, all of you, for being here. Yeah, before starting, I want to thank my collaborators with which this work wouldn't be possible. There is Seb, Pete, and Sri from the Broad Institute, and Lorin and Nava from Microsoft Research. So these works have been done with them. So what I will be talking about is one infrastructure challenge that we have seen in deep learning for single-cell omics, and in particular about data loading. And so I will present our solution, which is called SC dataset, to overcome this problem. And this, then I will do a small break if you have questions about this first part, or even interrupt me at any point. And then I will be talking more about a modeling challenge, and I will present hierarchical version of a cross-entropy loss for cell type annotation. And then after that, I will leave the stage to Lorin that will talk, among other things, about how the size of the data and the type of data can influence the training of deep learning models. So why deep learning? And I think the reason is we have seen in the recent years that deep learning is revolutionizing any field. For example, computer vision and now natural language processing. I think all of us, it's basically using a chatbot at a certain point during the day. And we have seen deep learning also revolutionizing biology. We've seen with AlphaFold and now with bolts, protein structure prediction, protein binding, and all this kind of stuff. We've seen that we have models that now can process the whole DNA sequences. And so the natural question is, can we expect the same stuff on single-cell omics? In particular, we will talk in all examples about single-cell RNA-seq, but most of the things will apply to any kind of single-cell omics that you can think of. And the real question is why we don't have something so powerful on single-cell omics yet. And I will try to give two answers to two of the problems that we might have in this field. And so the first one is what I call an infrastructure challenge. And to talk about this, there is a brief background, which is on the fact that we will be talking about Python and PyTorch, because this is basically the standard for deep learning. And also we have a good suite of tools for doing data analysis on genomics data and on computational biological data. And so we have ScanPy, SCVI tools, which is a suite of probabilistic models. So Python is a really nice scenario. And in particular, the developers of this consortium of tools have developed this data format, which is called the undata format, that is very handy to have annotated data. So you have the X matrix, which, for example, in scRNA-seq represents the expressed genes per each cell, and then you can annotate with information about the cells, information about the genes, and so far, so on. And it has some nice properties. It supports like sparse cell-by-gene matrices, which is usually the case for single-cell RNA-seq data. So we will have just a very small portion of genes measured during the experiment. It supports on-disk datasets, which means you don't have to load everything in memory when you want to access your data. And this is actually pretty useful for a very big dataset. And it supports sharded dataset, which means you can have different files, which are easy to distribute in different systems. And then you can concatenate them without the need of recreating a new file and like waiting for merging this kind of stuff. And if we want to do deep learning on this kind of data, we actually have just one native solution that works on top of this undata object, which is called the unloader, which was developed by the undata team. And the problem with this solution is that it's extremely slow for deep learning application, to the point that basically all the models you can use on them consume data faster, so process data faster than the time you take to fetch samples from disk. In particular, if you want to do like random sampling from disk, which is usually the case for deep learning applications. So we can ask like a couple of questions saying, okay, what could be like the solution to this problem? This is basically how we thought about the problem and we went through it. And the first question is, okay, what if we load the entire data set in memory? The problem with this solution is that nowadays we are getting with very big data sets, like the last perturbation data set, the biggest one is the tau 100 million data set, and like it will take several like terabytes of RAM to do this. So maybe some like data centers can do this, but if you want to do this on your laptop, you definitely cannot do this. So this is not a viable solution for like small labs and people that want to try this stuff on its own. So the second idea is, what if we stream the data from disk? Because like instead of sampling, what we can do, we just take contiguous cells from disk, which is actually faster than sampling from disk because of reasons that we will talk about later. The problem with this is that I will give as an example like the tau 100 million data set. But for this data set, we have that the data set is plate organized, or in general, when you do an experiment, usually the data that you have out are organized by the experiment. And so in this case, what happens is that each plate is composed of about like 7 million cells. And if you train this in a streaming fashion, what happens is that basically the model will just remember the last plate it was trained on, because we have a catastrophic forgetting, and so it will erase all the information before that. And we will actually see this happening later. And so the third question could be like, what if we change format? Like we've seen the undata format is pretty nice, but what if we change it? And we have a bunch of different reasons not to do that. The first one is it's pretty costly to do this conversion. Like if we are getting a very big data set, it could take like several days and several time to do this kind of transformation. And the second one is that we usually increase the storage usage because undata was like thought to be used for this kind of sparse data. And for example, if we take the tau 100 million data set on undata files, we get more or less 300 gigabytes of data. If you transform it to the Hugging Face dataset version, which is also available online, it's more or less 2 terabytes of data. And sometimes you have also peak storage usage, which is much higher during the conversion. And even if we do that, usually we still have slow random sampling because this is how the disk work. But since you're basically doing the conversion, some people just say, okay, let's do also a pre-shuffling while we do this conversion. And so some people solve this problem like that. And finally, like if we want to do some analysis after this, or like if at any point we want to do some transformation, like normalization of the data, we have to go back to undata format usually, because like all libraries works on this kind of format. And yeah, just last to mention, it doesn't work with many tools. For example, if you want to do probabilistic inference with SCVI tools, usually it accepts undata format. It doesn't work with other formats, or at least you have to wrangle around that. And so like the question we were left with is, if we rethink the way of sampling from the disk? And what we actually want is like a fast way to do random sampling from disk, because this is usually how SGD and other strategies works. But actually, if we can do fast quasi-random sampling where we will quantify this quasi-randomness, this is actually pretty good if we can get good performances with a good speed. And to do that, I will present you 2 strategies, which are called block sampling and batch fetching. So to start with, I will make just an example of like a training procedure. Usually, you have your model and you set up a certain mini batch size. Let's say that this is 64, which is just standard size for training. This means that in like normal setting, you will be sampling 64 cells from 64 different locations in the dataset. Now, if the dataset is on disk, this means that you have to do like 64 random accesses to disk, which is pretty costly. So what we thought about it, we thought about it and we introduced this block sampling. And what we do is basically we define a block size, and this block size tells us how many contiguous cells we want to sample from disk, for example, 16. And now if you want to create a mini batch of like 64 cells, you can sample from 4 different locations, taking 16 contiguous cells from this. And this is just basically a trade-off between the disk throughput that you can get and actually the diversity of the mini batch that you're getting. So not much in here. But now we can combine this with this other idea, which we call batch fetching. And the idea is that we can ask the disk for more data at once. So instead of like 1 mini batch or 1 cell, we can ask more than that. For example, we can ask for 10 mini batches. And why we want to do that, I will give you 2 reasons for that. So the first reason has to do with disk optimization. And in particular, the disk can do can serve batch requests in an optimized way. And to give you a sense of what does it mean, I can give you an example. It's a simple example with an elevator. This is just one of the many optimizations that a disk can do. For example, let's suppose we have 3 people that have to take an elevator. And the first one has to go to floor 2, the second one to floor 8, and the third one to floor 5. Now, we can serve those people sequentially. And what happens is that we first go to floor 2, then to floor 8, and then back to floor 5. Or like even if we can take 1 person at a time, we have to go back to floor 0 each time, which is even worse. And with this strategy, we basically travel 11 floors in total. Now, if the elevator knows that he has to go to all these 3 locations before, what he can do, he can stop at floor 5 middle way and like serve all 3 people. And now we just traveled 8 floors. And there is basically a similar idea for disk. If you know all the data that you have to access, it can optimize the way he accesses and serve you the request. And the second reason is what we call in-memory shuffling. And the idea, as we said, is, for example, we want 10 mini batches worth of cells instead of 1. So we are actually asking for 640 cells at a time. And we are still using the idea of like block size of 16. So now we are accessing 40 different random locations in the disk. But now that we have the cells in memory, so like we suppose that we have enough memory to handle like 640 cells. Now we can shuffle the cells in memory, which is pretty cheap because everything is in memory. And we can rebuild the mini batches after that. So now each mini batch, instead of having cells coming from 4 locations, we'll have many more locations, like 35, 36, depending on like how many mini batches you choose to start with. And so what we're actually doing is basically trading the memory consumption of your computing system for an increased throughput for the batch request and for more mini batch diversity with this in-memory shuffling. And this is basically the full picture of the whole algorithm. We have a certain data set. We generate just the indices of those data sets, which are integers, so they can stay on disk, in memory. We can then split in blocks that we define with a certain block size. We can shuffle them, and now we're shuffling just the indices, not the actual data set. And we can reconcatenate them in a single vector, and then we can split them based on the size that we want to fetch from disk, which we call fetch factor, so mini batch times fetch factor. And then we can just ask the data from disk, shuffle the resulting data in memory, split in mini batches in memory, and return those mini batches. So this is the whole algorithm. And then the question is, okay, how does this algorithm perform? And this is basically a plot of the throughput of the algorithm compared to like the choice of block size and fetch factor. And on the bottom, on the black dotted line, you can see unloader, which is more or less 20 samples per second, which is not that much for deep learning application. And like in this plot, if you go up to 64, a fetch factor of 64 and a block size of 64, we can get almost 1,000 samples per second. So this is actually 48 times speed up in single core setting. Now, the other thing is that unloader doesn't support very easily multiprocessing. You have to like wrangle with the source code. While in our solution, you can use multiprocessing in PyTorch data loader. So we also compare to that. And we have basically more than 2 orders of magnitude increase. And what does it actually mean, for example, for the tau 100 million dataset? It means that just to go through the dataset without any training, with Unloader, you will take 2 months. And with this solution, you just take 11 hours. And this is without training and anything else. And like previously, we talked about this quasi-randomness in the process. And we quantified this with this term like mini batch diversity. And we did 2 things. The first thing is compute this in a mathematical way, which is mini batch entropy. So what we did is, again, considering the tau 100 million dataset, as we said, this is plate organized. So we have 7 million contiguous cells per plate. So we basically took the plate labels of each cell, and we measured the average entropy of the mini batch that we were creating. So mini batch of 64. Now, if you do a standard streaming strategy, basically you will take the cells coming always from the same plate, except for like 13 times on the change from one plate to the other. So the average entropy will be 0. While if you take random sampling, we actually computed like the average entropy of random sampling with mini batch size of 64, and you get a 3.63. And now what we did is, okay, let's compare our solution with these values. So you can see on top the performance of random sampling. And you can see on the very bottom the performance of basically streaming, because this is not really streaming, but is a mini batch size of 64 with a block size of 64. So we are taking 64 contiguous cells. So that's, again, a batch entropy of 0. But you can see that as we increase slightly the fetch factor, we can actually achieve almost the same batch entropy that we will get with random sampling. And yeah, and this is just a mathematical way to like quantify this. It's completely task agnostic. You can validate this with any dataset, but we also try to do some real world evaluation of this. And we did it on the tau 100 million dataset. So what we did, we trained a simple linear model. We didn't want to like have confounding effect from like hyperparameter tuning, the choice of the model, or anything else. So just a simple linear model. And we evaluated the F1 score on 4 different tasks, the simplest one we can think of. Cell line classification. Oh, just to mention that tau 100 million dataset is just this perturbation dataset where you have 50 cell lines, 400 drugs. Then you have also dosages that we ignored, and you have all combinations and see how the perturbation perform. So we basically just took like the siRNA-seq of each cell, and we classified the cell line. It was coming from the drug, and the mechanism of action, which is basically a broader classification of like the drug effect. And this was like the labels were done by the authors of the tau 100 million dataset. And those are the performances. So we actually compared 4 different solutions. The first one is streaming that we presented. The second one is streaming with buffer, where buffer here means we can also have a kind of buffer as we presented for the in-memory shuffling for the streaming strategy. It's just that all cells will come contiguously. And this is actually a solution that some libraries do. For example, Hugging Face have a streaming with buffer strategy. So you can shuffle locally the cells, but actually we know that if we don't want this effect of plate, we will need a buffer of 7 million cells or more. So this is just a buffer, I think, of 1,000 or 10,000, I don't remember by heart. And then on the far right, we have the random sampling. And then we have one of our solution, which is block size 4 and fetch factor 16, which is actually the solution where before we presented an improvement of 2 order of magnitude. So we can even go higher than 2 order of magnitude, but we will get less performance. So we took basically the best solution from both worlds. And you can see that, like, regardless of the actual performances, you can see that the streaming and the streaming with buffer basically go hand by hand. And then we have the random sampling that goes well with our solution. So those are the pairing of the performances. And yeah, so we put everything together in a package. You can access the GitHub on the right with the QR code. We called it SC dataset. And this is basically a drop-in solution that can go on top of any collection. Like, we thought about it for the undata format. It actually worked with any collection. You can work with Hugging Face. And there is also this NVIDIA BioNeMo, which is another solution from the NVIDIA team to work with undata format. And the way we went to work with any solution is just we expose for methods that basically give you the opportunity to index your dataset however you like. There is a function called fetch callback and batch callback, and then you can transform also the data on the fly. So you can define everything you want for the dataset at time, and you can install with standard pip. So pip install SC dataset. And we also compare this solution to those other loading solutions. So the to Hugging Face, where like you can get the tau 100 million dataset on Hugging Face, and then to this NVIDIA solution that was specifically meant for like single cell data. And so on the right, you can see that basically the standard throughput is 20 samples per second for a loader. Then you have 8 times speed up with Hugging Face over unloader, and you have a 12 speed up of BioNeMo over unloader, while we get a 48 speed up. But as we said, this can be used on top of any collection, so you can even combine like Hugging Face with SC dataset and get even more. It's just that you will lose a bunch of things. For example, in case of Hugging Face and BioNeMo, those are not on data native. Like we wanted to maintain the compatibility. And also like the handling of metadata is much harder to do with Hugging Face and BioNeMo, while for unloader, also for our dataset, you can actually return undata object, you can return PyTorch tensor, you can return whatever you like for your task. And like now we support also weighted sampling. That's something that a lot of people asked for. So you can also train on imbalanced datasets, and you will get the same performances you get now. Yeah, so just to recap, like we wanted to handle this problem of like having data loading, like handling the data loading bottleneck for large scale deep learning. And so we think about this. We propose this SC dataset solution. So now you can train on this large dataset on your laptop, basically. And how we went for it, we basically trade pure random sampling with quasi-random sampling, and we show that basically with if you choose the parameter well, you basically get the same performance of random sampling, but you have 2 order of magnitude improvement. So I think this is a good time to stop if you have any questions about the first part. Yeah.
Hi, nice to meet you. Great talk. It's really, really interesting. So I'm from a pharma company. We worked on a lot of data sets, single cell data sets coming from clinical or real patients. While, and it is great that you can actually get to load sort of very big data sets that we're getting. However, I'm just slightly concerned whether the way that you quantify entropy assumes a level of sort of diversity between the plates for which has been tried to be experimentally controlled. So the tau data set, they're trying to experimentally make it as similar as possible with the least plate effects. So have you tried in a data set where it has patient samples in which the batch effect between patients will be kind of a very different distribution? Do you think that the ability to use the quasi-random sampling won't make the batch effect worse?
So we didn't try it on any other data set because like we did the setup for this large scale data set. I think that like even in this data set, even if it was controlled, there was some technical artifact because we can see the difference in performance. If this artifact is even more pronounced, what one can do is just like increase, for example, the fetch factor, and so you will use more memory, or you will slow down a little bit the throughput, and like you will handle this in this way.
Would you suggest sort of quantifying the entropy factor before sort of addressing? So
exactly, like it's very easy to quantify because it's just a mathematical formula. We use the label entropy, like the plate label entropy, because like we knew that the dataset were organized by plate. But if you have any other way of organizing your data, maybe by cell line, then you can use the cell line label or something like that. It's just that it's not super easy to like find a universal way to measure like how quasi-random you are doing. So it's either on downstream task, which depends on the model and the task, or it's with a mathematical formula on some labels of the data.
Thank you.
Yeah, thanks for the talk. In my experience, the bottleneck was always GPU calculation. So you of course, everyone always monitors their GPU usage while training their models, and using multiple workers per GPU. I didn't find it hard to keep the usage at 100%. So I don't know why I have a different experience. So that data fetching was not a bottleneck for me. Related question I have is, in your test in the Tahoe data set, did you measure real time of the training of the model? Like in these bar plots you showed with random sampling, block fetching, and streaming.
Okay,
so what's the difference in time?
So this was just about the loading of the data. We actually have tests on like training a model on top of them, and the time that doesn't change, because like in our experiment, the GPU basically had 0 utilization most of the time. So even if you put on top a training of the model, just get the same performance.
I see, because the model is small.
Yeah, we didn't use like a very large big model. Yeah, that's also the case. Also, just to add on that, I think it also depends on the server that you're using, for example, because we have been other people where instead of having a baseline of 20 cells per second, they had 200 cells per second. So maybe 200 cells per second with a very large, I don't know, foundation model, the transformer model, maybe it can work like that. So it depends also on the system you're in. But for example, in a laptop, it's pretty hard that you will get this.
On a laptop GPU isn't enough to train a foundation model.
Yeah, I mean, we will also talk about like, do we really need foundation models or stuff like that? So this is just a way of like, if you want to experiment with some models, even if it's not that big, you can do it. And even with bigger models, we don't want the data loading being the bottleneck. So like in our cases, if you want, I don't know if you trained on data format or you change format.
I used ndata, but I didn't use nloader. I always write a custom data loader in PyTorch.
Okay, yeah, that's also another
solution. And then I use multiple workers per GPU, set it to 4. If it's not 100, set it to 8, and then it always
yeah. So like what we did is actually we wrote a custom solution, but since basically every developer is now writing its own custom solution, we said, okay, let's make this accessible for everyone.
That makes sense.
But yeah, you had to write your own data loader. Makes sense now. Okay.
So which type of disk did you test? Did you use in this test?
Sorry,
experiment? Which disk did you use in this experiment?
Oh, this is basically a RAID of 5 SSD. So this is done on solid state disk. We didn't try on our disk because we didn't have them on the server. We expect even larger difference in performance because their disk actually have a physical latency. So this is actually the worst case scenario to show the improvement.
Yeah, and why, second question, why, so I didn't understand why you get a speed up when you turn on like parallel computation. If this is IO bound, why does it give you an improvement?
Oh, because probably we can, probably because we can go, where is it? Like probably we can go even higher with them, and so you don't have to use multiprocessing. Like if you increase even more the block size and you increase even more the fetch factor, probably you can go even a little bit higher. So that's, and probably you will get this. Like you can get this on single core if you increase this number enough, probably. The problem is, for example, for the fetch factor, this will actually use a lot of memory. In the tower, we have 6,000 measurement for each cell. If you have a batch size of 64, then it's 64 by 64 by 60,000. So if you start getting higher, you will use a lot of RAM. So there is also this this issue. But yeah, I think that in single core you can get it there, like doing this stuff. The other advantage of using multiprocessing is that, for example, PyTorch use this prefetching. So you have multiple workers that prefetch and organize your data. If you have a transformation that takes time, whatever it is, like it will do it in parallel. So there is also this thing. We basically, with this number, we iterated like also the CPU bound of doing this operation. Okay, I think we can move on and then leave other questions for the end. Okay, so now I want to talk like about one modeling challenge that we encountered and like our little success story on that. Like the general idea is like we really want those foundation model or single cell omics data or on biological data, but so far we didn't see really like good models on that, in the sense that there are a lot of works showing that linear baselines or some very simple model actually outperform those very big foundation model trains for days. And so we asked the question of why this is happening. And one of the many answers that you can get is basically we want to actually harness the structure of the biological task we're trying to model. And the problem with most of these models is that they are like at least at this like 2 years ago, they were blindly inherited from other fields. Now we are moving towards these more biological meaningful models. And for example, we can take the transformer architecture from like NLP, and like if you want to do an autoregressive framework, what you have to do with genes, you have to order, like you have to give an ordering of the genes that you usually don't have, and then you maybe predict like the next most expressed gene, but this is not actually the way you would model the task. So we actually want to build model that harness the structure. And we took this idea and we applied to a very simple example, which is cell annotation, which is actually a downstream task that most people do like to test if everything works out. And also most labs want to do like to annotate their cells. So what is cell annotation, in particular, automated cell annotation? The idea is that you have a data set, and as we said before, we will think about scRNA-seq measurement. And you want to put a label for each measurement of some kind. And like to do this in an automated way, what we can do is we can use some deep learning model to do this. And so what we will consider now is Atlas scale cell annotation. And in particular, we will focus on the Celix gene atlas. And in this case, basically what we have is this wonderful work that tried to basically benchmark a lot of different methods on this annotation, which is called SCTAB. And what they did, they took their own model, which was the best one, which is called, it's basically a different version of TabNet, and they compared to a lot of different models, a linear one, an MLP, and also like some foundation model fine-tuned to do cell annotation. And so when we've seen the results of SCTAB, basically two things actually catched our attention. The first one is that the difference between the linear model and the TabNet model is basically 4 percentage points, which is very small because we are not even like near the 1. So this task is not even solved. We are around 0.8. And there is this little difference between a simple linear model and the transformer architecture, which was their best performing model. The actual linear model also outperformed like some foundation models fine-tuned on the task. So that's the first thing. And the second thing is the way they did the split, which we called in-distribution test set. So what they did is basically they divided the data set in different patients. So you partition for patient, you will have some patient in training and some patient in testing. Now, if you want to do annotation in a real lab, what will happen is that you will have your own data set and you will annotate all of it. You don't train on a portion of it before. So we actually wanted to consider a more like realistic scenario, which we called out of distribution test set, where we actually took whole new studies to like annotate. And so what we did, we kept the original data set that SCTAB used, and we added a bunch of studies. Now, those studies was chosen with the same rules of SCTAB, so we have the same sequencing technology, and we have a subset of the original cell types because we didn't have the chance to cover all of them. And so we have 21 new studies. And like we basically found out two things. The first thing is that the performance of every model dropped a lot, like from 25 to 32%. And the second thing is that in this scenario, the linear model actually outperformed the TabNet model. So like we also seen before with other papers, like those very big models doesn't seem to perform really well when you test them in some scenarios. And so we actually asked, like, why is this the case? And to understand this, we first need to understand how we do the prediction and what is considered a correct prediction. So in this cell annotation task, what we have is basically we have a cell ontology of labels. And what happens, especially for Atlas, is even if those are basically harmonized, is that one lab maybe put a more broader label on a cell, and some other labs put a more granular label. So some labs maybe it's not really interested in lymphocytes and specific lymphocytes, so you just put lymphocyte, and some others like distinguish between T cell and B cell. Some others like for CD4 positive T cell, CD4 CD8 positive, and so on, so far, so on. So we actually have this kind of hierarchy that we want to learn on. And so let's make an example. Let's say that we have a true label given by our dataset, which is a lymphocyte. That's all we know. Now, what we can consider a correct prediction from a model that learns on this kind of hierarchy. Of course, if it predicts that it's a lymphocyte, this is a correct prediction. Now, if it predicts any of the subtype, we have to consider this right because we don't have the knowledge to say it's wrong because we just have the more broader prediction. And anything else is basically wrong. If we predict something broader or something different, it's just wrong. So this is basically the setting we're into. And so what we actually want to do is learn on those direct acyclic graphs that represent the structure of labels. In particular, those labels are basically set inclusion. So you have that when you have an arrow, you say that you have a subtype, which is a subtype of the parent node. So we know that annotations have different granularity, but what have been done so far is that we treated this task as a classical classification task. And so we trained it with cross-entropy. Even like in this study, they trained it with cross-entropy laws. Yeah.
Really quick question. If you could go back to the hierarchy there, you note that you don't like you consider leukocyte to be the more general label to be incorrect, but the more specific label to be correct. And I just want to note that I think that I've had to deal with this problem myself, and I've had to come to think to myself, like, when do I think actually a more general or a more specific label is more correct or not? And I could imagine a situation where if you don't know a specific label, you would prefer something more general, where if you label something a T cell and it's actually a B cell, that might be worse than labeling it as something more general. So I was curious if you could comment on, that sort of value structure.
Oh, yeah. So, there are a couple of reasons. The first one is that also, for example, SETAP did it this way. And the other reason is that you have a model, and the model will just take RNA-seq sequence, and it will have to give you a label. Now, this label that it will give you cannot depend on the label that you've put on. Like if it is a T cell, and it knows that it learns that it's a T cell, you may have end up with a lymphocyte because you didn't want it to go deeper, but the model cannot know that. So from the classification task that we're trying to model, we have to give the benefit of the doubt to the model because we don't have a more specific label. The other solution is just basically throw away all the broader. But what we will actually see is that even if we focus just on the leaves, we actually get better performances with this method. But yeah, that's a good question. Thank you.
I think that what they're implying is that sort of, and this might be another step, but you might want to penalize the wrong label in a different way than just wrong and right. So from a biological standpoint, again, it's not part of this, but it's essentially if you can insert penalty in whatever training you do, there are biological penalties that you can sort of see who's worse than others. That's all.
Oh yeah, actually, this is the point of what we want to discuss. So this is just how do we evaluate things. So this is basically how do we compute the accuracy of the system. Now we will discuss how do we penalize the model to do that. And basically the standard strategy is that you treat everything independently. And so if you have a T cell and you're predicting it with a B cell, would be considered wrong. But also if you have a more specific subtype of a T cell and you predict that, but your label is T cell, would be considered wrong. Because in a classical cross-entropy loss, you don't have any kind of structure in the data. So for the model, everything is independent. And that's basically the problem that we're having. So our solution to that was actually reflect the kind of structure that we have in the evaluation task. So if you now look at all the nodes, you don't have a real probability distribution anymore. But what we wanted to achieve is basically if you have some kind of probability scores on some nodes, then your parent category needs to have at least the same probability distribution. You can make it equal, or you can make it at least having a little bit of room for something that you haven't seen or something else. But basically, this is the strategy. So we want to make sure that if we put 0.5 probability on gamma delta T cell, then we have at least that for a general T cell. Then when you train with this kind of objective, what happens is that the interpretation of the vector you are getting is different. Like you are still predicting over the whole, all the nodes. But now when you look at the row probabilities without disaggregation, the interpretation is that if you have some probability over a T cell, you should interpret this as this is a probability of being a T cell and not being any of the child, because otherwise the model would have put probability on that. Well, in the classical way, you cannot give this interpretation. I will actually show you an example of synthetic data. You can actually follow the notebook on the link. You can download it, do whatever you like. So we just come up with a random custom directly acyclic graph. We make sure that it wasn't simply a tree. And we have 5 leaves in this hierarchy, 4 on the same line and 1 slightly before. And basically what we did, we generated some synthetic data where basically each spot corresponds to 1 leaf. And then what we did is basically the color assigned to each cell was either the leaf or any of its ancestors. So the actual generation process is that each of those 5 spots correspond to one of the leaves. But we put the labels following the hierarchy. So for example, in one spot, you can have both the label of like the leaf 8, or even like its parent 4 or 1 or 0, but not the other. And what we did, we trained a very simple model. It's actually a 2-layer MLP, the simplest you can get, with a cross-entropy and hierarchical cross-entropy. No hyperparameter tuning. Like it's just like I think 100 parameter model, something very easy. And what we get out of this is with cross-entropy, you get an accuracy of almost 0.6. With hierarchical cross-entropy, you almost get 1. But the cool thing about the toy example, we can actually easily visualize what does it mean in the 2D space. And so what happens in the cross-entropy case is that since all labels are basically independent one from another, the model gets confused and doesn't know which label assigned to each spot. So maybe if you get more of one label than the other, probably this will take the whole spot, since the model is very simple. You can go more granular, but basically you will never be able to like assign the correct probabilities. While in our case, what you will see is basically each spot get the label corresponding to the corresponding leaf. And the reason for that is that if I give in the model a high probability, for example, to the leaf 8, I'm also giving the same probability to the leaf to the node 4, 1, and 0, because we are up. Yes. Okay, because we are basically up propagating this probability. So now the model doesn't get confused anymore, and basically almost every prediction falls down to the leaf. Okay, and now now we can show some results on the actual like Atlas scale data set. And so first of all, those are the results of 3 models. Those 3 models are deep learning models that are trained end-to-end from the SCTAB study. We kept exactly the same upper parameters that they found optimal. We just changed the loss. We didn't run another hyperparameter optimization. So those can be also suboptimal as results. So you can see on the gray bar the cross-entropy performance, which was the one dropped by basically 25 points. And then on the right, the hierarchical cross-entropy performance, which is basically 12 to 15% more performant than the corresponding cross-entropy loss. Then what we did, we also analyzed like how does it, like this is basically a bulk measurement per model. We actually wanted to see how does it work, for example, for each cell type. So those results are for the MLP model. This and the next one are all for the MLP model. We have very similar results also for the other models. So what we can see is basically, so those are the 80 cell types that we found in the out of distribution setting. And every significant improvement is basically, every significant change is an improvement over the standard cross-entropy. We just found one of 2 cases for the linear model where there is a significant, like statistically significant decline, but we also have to say that the linear model is very simple. But for the MLP and the tablet, every improvement, every statistical change is an improvement. This is for cell type, and also we looked at the at actual studies, since what you want to do is just give you a new study. And so we wanted to see if for each study we actually get an improvement, and that's also the case. Like every statistical change is actually an improvement over the previous result. And I think that's it. That's a summary. You can look at the paper on the QR code. And yeah, like we propose this hierarchical cross-entropy loss, and we've shown that this improves out of distribution generalization on every cell type of any study. We also have some other plots on like tissues, disease. We basically have uniform improvement over any kind of categorization you will get. And also this idea can be extended to any scenario. There are not many scenarios out there outside of biology, but if you have some labels, even for like image classification, where you have this kind of set inclusion relationship, you can also do that. Like if you have a label that is an animal, then you can do this kind of play with like cat and dog belonging both to animal. And this is in general a big opportunity for designing new architecture and training strategies for biological tasks, because we have shown that no matter the architecture you choose, like a simple linear or more complex transformer, you basically get improvement if you model the task correct. And I think that's it. Yeah. Thank you very much.
Thank you, Davide. Time for questions.
Hi again, great work. And I'm very, very happy that people are starting incorporating biological sort of thinking and how they actually model it, because it will make a huge difference. I guess I have one good question about sort of your comment about the fact that you can only reach about 84% accuracy. One of the issues practical issues that we're having is that, when you define a ground truth, when you look at a cell axis, it's like somebody annotating that cell. So you're basically using the ground truth on partial truth, and like it's literally partial truth. So I even doubt that you ever get 100%, because you'll have to sort of verify multiple wrong at the same. So that's one thing. And two, I think we practically speaking, we actually use a signature-based non-AI model to annotate the cells, in which you basically use hierarchical cell annotation, which basically means that every cell that we annotate doesn't have one label which is flat, but actually has like a treed annotation.
Yeah.
Is there any way you can train on that, first of all?
I think it depends on how do you implement that. I think that in SCTAB paper they compare to cell typists, which I think works like that. I'm not sure, but I think those are basically a logistic regress, like a tree of logistic regressors. So you do this kind of job, and they show that basically they outperform also that in their study. I mean, one can think of training or something like that. Depends on how you define the model, I guess.
And secondly, I know this goes out into biological expertise, and maybe this is outside of your realm of interest. But for example, one of the sort of improvement that you show in the last was like T17 cells, if I saw correctly. And I think, from an application point of view, it'd be really, really interesting if you're actually able to prove that those cells have the phenotype that you're looking for. So that you kind of run an experiment in which you do single cell sequencing, and you do cytokine labels or cytokine measurements, and then you run and you try to identify them and see whether you can actually tell apart like the functional state, which is what's really sort of hard to define in these very sort of fine-grained sort of T cells and B cell states. Yeah. Anyway, great job.
Yeah, thank you for the suggestion.
Over here. Yeah. Thank you so much. This is a very, very cool topic. Some of what we've seen in the lab is that it's very hard to annotate cells that have very low cell numbers, especially with the cross-entropy. Do you see the biggest improvements here in the performance of the model in those cells that have very few numbers in the training set? Or have you looked at that?
So you mean like rarer cell type get better improvement?
Correct.
Mm-hmm. I think it's, we had some plot, I think it's pretty independent on the rarity of the cell type. It has more to do, so basically when you have this hierarchy that is basically derived from like the cell types that you have. It's just basically you take the cell ontology and then you take the subgraph induced by those cells. It actually happens that some cells are disconnected from this graph because you don't have maybe the parents in the annotations. So we saw the biggest improvement where you actually have destruction. Like for example, for the isolated nodes, you don't get much improvement because it doesn't make really a difference because you don't have this this hierarchy. But for other things, I would say it's pretty independent, for example, from the rarity of the cell type.
Awesome. Thank you.
Do you think if you trained on a mixture of, let's say, coarse and fine labels, but then only evaluated on the finest grain labels you had, you would still see the same lift with the hierarchical loss?
The same what?
Would you still see the same boost from the hierarchical loss compared to the standard?
Yeah, I would say so. So we also have like the kind of plot of the of like 2D plot of how the cells are organized. As I said, this is also independent, mostly independent. I mean, there is a little bit of skewness, but mostly independent from leaves or internal nodes. And basically, as you can see, we have, where is it? We have basically an improvement over all cell types. This also includes the leaves, which are most of them. Like half of them will be leaves. So yeah, I don't know if it would be exactly the same, maybe 2 points less, 2 points more, but basically you will see it.
The reason why I asked is I'm thinking about the case where your label is lymphocyte in the training data, and the model predicts T cell, but the true cell is a B cell. And they can become overly confident in its predictions at the finer grained labels. I could see that maybe hurting performance at certain levels of the hierarchy at test time.
Yeah, but like, what's the alternative in the sense that, like, with the cross-entropy, even if it doesn't get like overconfident, if it predicts the broader one, it's still not correct from our point of view, like from the class.
Yeah, but in its sense of what the finer grain labels are, those points end up becoming like noise, right? Because it predicts a T cell and it thinks it's correct even when it's actually a B cell. So that test time, if you're only focusing on the finer grain cells, then I could say maybe being less certain about the finer grained classifications. I'm not sure.
I can see the point. I would say, like, what we have observed is that, like, on specifically siblings, usually you get better performance.
Okay, interesting. Okay, cool.
Yeah.
Any other question for Davide?
I actually have one. You mentioned briefly that you said the linear model showed, I think, maybe the least improvement or something like that. And I was curious about that because one of the things that I like to do sometimes is I like to have a linear model for things because you can just check and see what it is. Like, why does it think this? They're very interpretable. And I was, so I like to try and give them some credit whenever I can. So I was curious if you could comment on, the failure mode there a little bit more.
I think we didn't really analyze that. So what you're saying is that, for example, in this case, the linear model without cross-entropy is outperforming the tabnet.
Or I mean, I think you mentioned at some point that you saw more declines.
Oh, yeah. So yeah, it's basically the same, but probably here it's easier to see. Yeah, like when you evaluate an out of distribution, you basically see that the tabnet drops 32% in performance, and linear 25%. We actually didn't analyze like the mode of failure, like we didn't check into the linear model. But like, if I have to guess why this is happening, is because the tabnet model can pick up like biases in the data more easily. So maybe pick up like lab-specific technical artifacts, and so this can be translated from patient to patient. While in the linear model, maybe you cannot do that. So in out of distribution, this drops performance more than the other. But we didn't really look into.
Right. Okay. So let's give Davide a round of applause.
Okay.
So I suggest we take a 5 minutes break and we start again at 10 with Lorin's talk. So yeah, see you in 5 minutes. So it's my pleasure to introduce Lorin Crawford, principal researcher at Microsoft, who co-directs a group here, also at the Broad, called Project Ex Vivo. He's going to tell us a little bit more about it. And today, the title of his talk is When More Isn't Better, Rethinking Scale in Single-Cell Foundation Models. Lorin, the floor is yours.
Awesome. Thanks, everyone, for coming and hanging out for the extra hour. So a couple things. I hope this talk is a little bit of everything, a little bit of like shameless plugs for our group, a little bit of internship calls for MSR, and then also science. The title is a new working title that I decided to use just for this talk. There's going to be some negative results, but I don't want you to take this as like I don't like foundation models. I hope you see it as a place for opportunity, let's call it that, for future development stuff. So for those who don't know, I'm a researcher at Microsoft Research. I always kind of start these talks, particularly in groups like this, because I don't know if everyone knows what MSR is. Microsoft Research is a global organization. We have labs stationed all over the globe. And you might ask, like, what is it that people at Microsoft Research do? And the answer to that question is like kind of a lot of stuff, almost like everything. there are people who are in, yes, computer science, but also people like myself in biology. There are also people like in quantum physics and things like this. I am part of what they call the BioML machine learning group, which is a subgroup of a thing called Health Futures, which is the life sciences and biological arm of Microsoft. People primarily stationed in Redmond, but also in here in Cambridge, and then also in real Cambridge, UK. And so the BioML team is kind of spread out across different both roles and also disciplines. So you have people who are both applied scientists, people who are researchers, and we also have an RA program for people who are just coming out of undergrad, which is also really nice. So what is it that we all kind of do? So, you know, broadly speaking, BioML kind of covers different scales of biology, if you will. You have people like Kevin Yang who are working on protein language-based models on this like kind of molecule stack. People like myself working in cells. People like Kristen Severson and Alex Liu working on this like kind of tissue level thing with things like histopathology, is also like things like microscopy. And then you also have people working on this kind of patient stack here. For myself, my background is I'm a statistician by training. I worked kind of on interpretable machine learning. My research program is kind of this idea of how do you understand how non-additive variation plays a role in complex traits and diseases. You can think about that in statistical genetics. So this idea of, you know, I can study how gene A's effect plus gene B gives rise to a trait, but also how gene A's effect times gene B gives rise to a trait. And can we use ML to kind of look over large search spaces to understand where these interactions occur or don't occur? I was also co-advised by someone in pharmacology cancer biology during my PhD at Duke. And so I've kind of gotten back to my roots and really thinking about this on this cancer stack. So thinking about problems in cell biology, very similar style of questions, this idea of now not gene A's affect gene N plus gene B on a complex trait for patients on the patient scale, for things like height and body mass index, but things like how do I understand how interaction between genes or pathways play a role in like therapeutic resistance? Okay. So a little bit of background about like what our group does, our group being this idea of like Project Ex Vivo. I'll kind of give a shameless plug about our group. You know, we're really thinking about ways to move beyond, you know, DNA-based measurements and really thinking about cell state as a marker for cancer therapeutics. So for those who may not work directly in this area, you know, DNA is kind of the cornerstone of precision cancer medicine, right? This idea of, you know, a lot of patient tumors have, you know, diverse backgrounds that are kind of measured by DNA via these patient alterations. When it comes to drug discovery, you know, we use a lot of patient models, cell lines, and organoids, where we study the fidelity of these models based on also DNA. And then when I think about how to stratify patients, right, I also think about this with relationship to whether or not a drug might be actionable or not actionable based on, you know, again, these like DNA mutations. And so this idea is that this pipeline sort of works really well, only if we think about the future that's relevant to drug response being DNA-based encoded, right? So, you know, how well have we kind of done in this paradigm? Well, there have been a lot of cases where this has kind of been a success, right? We're thinking about leukemias, breast cancer being one of these, but there are a lot of challenges in this space as we currently sit, right? There are only 40% of patients that kind of fall within this regime, where majority of patients don't have like an actionable single alteration, right? And the big challenge here is that, you know, we have limited responses due to both heterogeneity and or development of resistance that can happen both because of a genetic mechanism, but also something that might be non-genetic, right? And so we were asking these patients, like, how many of these dependencies can be explained by just like single individual features? And so, you know, if I think about this, you know, cancer phenotypes kind of have this really diverse set of inputs, right? You know, I just kind of gave this notion that we kind of see here on this DNA code. You can think about that as like this static mutated blueprint, right? What's really interesting, though, is you kind of move down this trajectory here, you kind of have this idea of, you know, RNA is kind of this dynamic interpretation of what happens within cells, right? It kind of captures in some sort of a tagged way or correlated way. You get some sort of idea of, you know, a little bit of measurement that kind of is correlated with what's happening on the DNA mutation scale, also things that are happening epigenetically, but also things that kind of interact with our environment, right? And so if I think about, you know, where we are, you know, you can kind of think about this idea of cell state being these groups of genes that are expressed by a cell that kind of capture both, you know, cell function as well as like an environmental phenotype. And so the current pipelines are here, but what we're asking in our group in ex vivo is really this idea of, can we search for state-based lethality on this via this cell state-like based measurement, right? And so for a little bit of background, again, shameless plug about our group, we have great tools to do this, but we're really asking questions like, how do we measure complex cell state attributes? And so ex vivo is a collaboration between Microsoft and the Broad, with a close collaboration with people at Dana-Farber, where we're really trying to bridge the gap between biology and machine learning with a diverse group of both experimentalists, computational biologists, and also AI and ML experts, right? And it's co-led by myself and Ava Amini at Microsoft, as well as collaboration with leadership from Peter and Sri. Pete's in this audience here. Just and a lot of lab members actually throughout here that are more than happy to chat with. We're co-located in terms of both geographic, but also like in space. So for those that don't know, Microsoft Research is actually down the street. I have no idea which way we're facing, but we're on one Memorial. And so we're not that far away. We also have lab space here at the Broad, both on the 5th and 6th floor. And I'm sure people like Pete and Sri and also Andrew and others will be more than happy to take you around that space if everyone's ever interested. And so ex vivo kind of has two broad aims, if I were to put them in different buckets. The first is this idea of what's the real fidelity of cancer cell states. One question that we kind of ask is like, how does the environment affect cell state and therapeutic response? And the other aim, which is a big part of this, is this idea of building foundational AI maps, or in these like kind of state to function maps. I'll get into that a little bit. And really asking this like fundamental question, which is like, can we learn a foundational AI model of cell state? Okay. There are really cool stories that kind of happen with both of these aims. Because it's the MIA talk, we'll just focus on this latter half. Right? So in this talk today, what we're going to focus a lot on is this idea of single cell measurements. So instead of thinking about a bulk, who are people that work in the space, instead of thinking about like an average expression readout from a given sample, we'll think about sequencing across individual cells to get this idea of like cell type heterogeneity, right? So for those who are more quantitative, you could think about this as like a large scale matrix where my rows are different cells, and then my columns are the expression values of those given cells, right? And so this idea is like, we want to build integrative approaches to measure and understand biology at this single cell resolution. Okay. Now, what's really cool about this era that we're in now is that, there was a point in time where getting single cell data was really hard to find, and you had different sources that were kind of scattered all over the place. Now you have these like atlas scale level efforts where you can get these data at high throughput. What's really nice is that this problem of working with single cell biology is both equal parts computation is also and also biology. So for the, again, more quantitative people in the room, some calculations that we used to do back at the envelope is like this idea of like, in terms of data points, if I think about this from like a multi-omic study, if I just were able to get data on DNA mutations for 500 cells across 225 genes, as well as thinking about RNA expression across 500 cells and all 20,000 genes, and I had some kind of idea of interactions across environment, your data space can explode quite a bit, where you could be working with like up to like 44 million data points like per biopsy. And so these tools that like Davide presented earlier are quite useful in thinking about, one, how you load data in at a scalable way, but also making sure that you don't just have to be at a place like Microsoft to make an impact in this space, right? And so with all this data sets kind of exploding in nature, one thing that people are really been started thinking about is like, obviously, like, how do I start to apply AI to these types of methods, right? Into these types of data sets. And so we'll kind of go through a little bit across like some of these AI different models, and then we'll kind of see what we've found in working with these methods in our own group. So just take a step back. This term foundation model has kind of popped up everywhere. For those who may not know what this is effectively means, foundation models are just effectively AI models that can do a diverse set of downstream tasks. So, very fundamentally, I might have some data set, I might train an AI model. You're going to hear me say these terms quite a bit throughout this over the next 45-ish or 40 minutes, is thinking about this idea of you split tasks into two different regimes. We'll start with the bottom, this idea of zero shot. So what we mean when we say zero shot is I take a data set, I train an AI model with it, I take that model, I freeze its weights. Its weights were just trained on a pre-training data set, and I apply it to something out of distribution or some downstream task without ever updating these model weights. So that's zero shot, right? I've trained it on something, and I try it downstream. Fine-tuning is a little bit different. Fine-tuning says I train an AI model, and then I take a little bit of data from maybe the task of interest. So I'll take a little subset of that data. I'll update my weights with respect to this data that has my task that I'm trying to do, and then I'll take that updated model and then apply it to my prediction. That's the idea of like fine-tuning, right? And you can imagine in a lot of different areas of science, where both fine-tuned prediction and also zero-shot discovery capabilities are wanted, right? And so, how do these models kind of work? So if you think about a model like GPT, just again, so everybody's on the same page, a lot of these use what they call a masked language modeling pre-training objective, okay? What that means effectively is I take a sequence, so let's say a set of text, we'll call this let's say learning about AI is awesome. What you'll do is you'll kind of tokenize this sequence. So effectively what you do is you'll find some representation with respect to an encoding for each part of the sequence. The masking part means I'll just go in and I'll mask part of the sequence, and I'll throw the masked tokens into my model, and I'll ask the model to then fill in the gaps, basically, right? And I'll judge how well it does based on some sort of loss. And so we heard Davide talk about earlier about this idea of cross-entropy, and I'll ask how good was the model at making the prediction and filling these given gaps, and I'll do this all over again. Now, the same kind of regime of the background of what's happening in all of our phones and things like this now is what we try to do is take the same kind of regime and apply it directly to single cell biology. We'll talk through why we think this is great or not great, but effectively you could do the same sort of thing naively with something with single cell. So I could take a set of genes and their expression values, the same sort of idea. I can mask a part of the gene and its expression, and I'll ask the model to kind of fill in the gaps with this given thing, and I'll ask the same kind of questions. How well was it able to do and fill in this thing of a given sequence? Now, naturally, if you're thinking about the difference between this setting and this setting is naturally, I have order and sequences here where you might ask this question, how do I think about how to nicely order genes? And again, these start to be modeling choices that people are starting to do, but naively, we don't necessarily have that nice structure to naturally think about how to do this well. So, where has AI kind of had a lot of success in our space? Well, in a few different places, right? And so if I think about different biological modalities, the first things that kind of come to mind are things that kind of have natural sequence-like structure in terms of their data, right? So if I could think about DNA sequences or protein sequences, there are a few things I want people to take away from this slide. One is this natural idea of going from text to sequences. It's nice to have this nice ordering of my data. There's another thing that's quite important is this idea of the reference training data sets. So one, if you think about particularly with protein sequences and you think about the success of something like AlphaFold and others, is I have both real data sets where I can think about where to pull millions and millions of sequences from, but I also can leverage synthetic data as well because I understand the biological processes that kind of generate nice sequences, and so I compare both real sequences and synthetic sequences to train models with high accuracy. So that's the first kind of thing is this training data set scale is massive. The second thing is that the underlying task and function are often tied to biology and things that I can also test, and also that people who are journalists really care about, right? This idea of in protein language models, the idea of can I generate models that can now identify new antibodies or new enzymes, right? In DNA language models, a lot of times we ask questions like, can you find enhancer regions around the genomes? Can you identify promoter regions, exons, so on and so forth? And so we had these really nice underlying function and tasks that kind of are also biologically driven. Single cell is not necessarily, or RNA expression is not exactly in this same exact space. We have a growing set of training data sets. We can have gene ontologies and gene sets. We have this idea of single cell atlases. We can leverage pathway information from different databases. And we have different tasks that people kind of do. So if you split out the different tasks, the three major-ish tasks that people think about, or I guess I left one out here, which is, if I think about fourth, is we can annotate new cell types, which is the idea of what Davide D'Ascenzo went through earlier in this talk. You have this idea of batch effect correction. So this idea of can I use foundation models to kind of integrate across different data sets? And you also wanted this idea of perturbation effect prediction, right? This idea, if I've trained a model on cells from one tissue, can I then predict its effect, right, in an unseen tissue or an unseen state, right? Or if I've trained a model on a given drug, can I predict in a tissue, can I predict the effect of a different drug, right, on that same tissue, right? And these are not necessarily the same as some of these. A lot of these analyses, as a lot of you pointed out in the last talk, is this idea of like, how well will I ever be able to do this when the idea that the labels that I now train on are coming from human annotated labs, right? And so some of these tasks are quite hard, and as a lot of people kind of point out, we'll get back to a little bit, this perturbation effect prediction question is like quite challenging, even though I think that actually might be like the gold standard that we're all trying to work towards. Cool, but despite all of these things, it hasn't stopped our field from trying. Like we've been trying. There's probably a new foundation model that came out this morning that I haven't seen yet, right? Like every single day, there's a new group that claims that we have a foundation model of biology. So I just picked 4 here. Sorry if I left off your favorite one. But there are a few things that I kind of want to get at. First is I picked 4 different models here that are all kind of different in terms of how they end up doing their model architecture and their training, right? The GeneFormer has a different scheme where it chooses the order and it sequences based on sequences of genes based on like the magnitude of an expression vector, and that's how it does its ordering. UCE kind of tries to leverage, it has like a universal cell embedding idea that leverages both expression data as well as data that comes from protein language models. Embeddings, SCGPT has a different architecture, SC Foundation has a different architecture, so the things like SC similarity. And every model claims to be foundational in the way that they do things in terms of performance. So it's hard to kind of, for those who are thinking about research, it's hard to gauge who's better than who also because not every model is publicly available. So like if you kind of have to take things at face value because people don't always release code to then end up going and retraining their models to really have this kind of really head-to-head comparison. The second thing that I think is like also quite important is this number here. It's the number of cells that each of these models are actually trained on. And I'm going to circle back to this, but I kind of want to put this bug in your ear. The first thing is that gene corpus for the gene corpus for GeneInformer was trained on 30 million. UCE was on 35 or 36 million. Here, SCGPT 33 million. SC Foundation was 50 million. There was actually a recent paper that was trained on over 100 million cells, right? So the size of these training corpuses continue to grow, and the size of these models also continue to get bigger, right? And so there's one kind of question I want to put in the bug in your ear, which is like, why do we keep doing this? But we'll get back to that in a second. So one thing that we started asking ourselves, and I'll take you through the story of how we got here, is a couple of years ago, we started asking ourselves, like, what is the actual zero-shot capabilities of these models? I can imagine if I trained a model on something and I fine-tuned it with respect to a given data set that was kind of aligned with the task I wanted to do, I can see a model doing kind of well there. But what is the model's actually zero-shot capability? If I trained it on a certain data set and I tested it on another data set where I could no longer fine-tune, like how good are these zero-shot capabilities? And so a couple of years ago, we had this idea of like, let's start asking these questions. And this was kind of led by an intern at our at MSR, Kasha, who asked these like kind of 3 fundamental questions. One, which is, do these models learn the distribution of data well? Do the embeddings capture desired biological variation from these models? And do these models from these embeddings correct for undesired technical variation when they come from different studies or different technologies? Okay. And so here's this first kind of result that we did that kind of started us down this path. So here's an example from SCGPT where we're looking at its pre-trained objective, where the idea is to predict unseen genes and its mass objective, okay? So this side is going to be where we do this when we look at just the training data set with respect to mass gene expression prediction. And here on the right-hand side, what you're going to see is the same thing, but we do predictions from using just cell embeddings, right? And ideally, if you were to do this really well, what SCGPT does actually is it takes, to make it that kind of discrete, it takes the expression of every data set, aligns them, and then puts them in these bins. And so what it's actually doing is predicting these bins. And ideally, what you would see is a nice diagonal, right? That if I was predicting well, I would have low bins for these cases here for the lowly expressed things, and it kind of scale up for the highly expressed ones. And what you're kind of seeing is not this at all. I'm kind of just, when you're just like kind of predicting this sort of mean, And here on this other side, if I do with cell embeddings, I'm not doing so well here for these locally expressed things and a little bit better for the highly expressed. So you might ask, like, how does this now translate to performance? And so here's an example of what we kind of did, where we thought about training data sets, where we wanted to predict gene expression using these, this different model. Here we're going to focus on SCGPT, trained on different types of data sets. The light axis is going to be mean squared error, so a lower number means better performance. The first model you're seeing is our baseline. So what we do here is what Kasia did here is she just took the mean of every single cell in my data set, and that's my model. There's no training, okay? And that's how well that model does. Now, what we could do is now I can say, what if I gave SCGPT just straight noise? Like I gave it like random numbers, like there's no real data here, and I trained it on just like random data. And I asked to predict, did you just gene expression reconstruction? Well, it does quite poorly with that when I feed it noise, which makes a lot of sense, right? I didn't actually give it any trained data sets. The rest of it actually is when I give the model actually real data. So what happens when I train the model on on cells coming from the kidney, cells coming from the blood, or cells coming from just humans, so regardless of tissue. And you can kind of see none of these models are that much better than me just using the mean, this like mean model as my predictive model. So then, okay, so that's not great. So then the question becomes, again, we're looking for opportunity. This is not a bad thing about these models. There's a chance to grow, right? So then we start asking the second question, which is, okay, what about the other kind of thing of annotation by cell type, or am I doing well of getting rid of technical variation? So for the first case, you can kind of guess based on Davide's talk how well this is going to go. Not fantastic for some of these models. So on the far left-hand side, what you're going to see is all these genes from this corpus kind of annotated by different cell types. And on here, you're going to see is a highly variable gene model. That's a kind of baseline where we take just the highly variable genes as our model and say, how well is this at predicting a given cell type? And you can see how well you can kind of do by kind of getting this nice clustering and this embedding on this UMAP of certain cell types together. Here we use Harmony, which is, again, it's like kind of data integration one. Harmony does quite well at this task. SCVI, which is a basic autoencoder, also does this very well. And then GeneFormer and SCGPT struggle a little bit in dissecting different cell types. Again, not surprising given our last talk. But you might have this question of what it is that GeneFormer and SCGPT are actually learning. So let's look at the same plot, but now let's color code by sequencing technology. So again, on the left-hand side, you have this idea where everything here is separated out by sequencing tech. The idea, if you're really good at integration, is you should look something like Harmony, where I have all these technologies kind of integrated together. You see how well Harmony does, and SCVI is not terrible at this task. And effectively, GeneFormer, as a stark example, is like just learning batch effects. So it's just learning sequencing tech. And so what we started to think about was, okay, well, out of these three things that Kasia kind of showed, you know, how well is this model like learning the distribution very well? Like it's not doing so great at that. Are these embeddings capturing biological variation like somewhat? And, you know, really these embeddings are just kind of correcting for, they're not really correcting for undesired technical variation at all. And so we put this paper out, and I was kind of nervous about it. But ironically, other people had also identified very similar things. Actually, people in this building on floors right below us, right? Where there have been a lot of papers since then that have come out, which have kind of noticed very similar things, right? That, you know, single cell foundation models tend to not do so well as linear methods, particularly if you think about perturbation prediction. I like this idea of one PCA that kind of rules them all. There's the recent papers that came out even this year in Nature Methods, this idea of like, I could just take a linear model and that tends to be a better model than having all these other type of methods. Actually, we'll circle back to the finding of this paper that we actually see in our studies as well. And there have been more papers that have come out since then, kind of around similar ideas. Now, if you think about this, one thing you could ask is, how do I fix the architecture of these models? And that's one question that we could talk about. But one question that kind of came to mind to me is going back to this data question. So I have this kind of reality here, but one thing I kind of asked is data abundance or diversity somehow bottlenecking these things? Like, is training data kind of the issue? Like, is it not the architecture what we're actually training the data on? you could think about this as idea of like, I like to think about this in 3 different parts where it comes to these different methods. You know, I have the architecture, which to me is kind of the car itself. I have the data, which is kind of the gas I put in the car, and then I have the task, which is like what I drive the car on in terms of road and challenges. And I really thought about this idea of like, are we putting the right data or right gas in the car to really make this thing go? And so we really asked this question of like, can we construct nice training corpuses to do this? And if you remember, I said that all these models were trained on 50 million cells, 30 million cells, 100 million cells. There was someone, I think maybe here, that said they were going to create a corpus that had 500 million cells. And it was like, whoa. But like, how much information are we getting per cell, right? And so I asked this question, and this was kind of co-led by a student of mine, Alan, as well as Maddie Hughes and Akshaya, who's now a scientist here, where we asked this question of, can we kind of think about how much these data kind of saturate or kind of like scaling laws with respect to data in these given corpuses? So what we did is like 3 different experiments that are a lot more experiments, and I'll show you a little bit. But the idea of this was, if I took data sets from SCTab, so SCTab has 22 million cells, and I downsampled them to a fraction of their sizes. So I took all 22 million cells and I downsampled it to have 1%, 10%, 25, 50, and 75% of its data. And I do this in 3 different ways, right? The first way I'm going to do is just uniform downsampling. So I'm going to take all 22 million cells and I'm just going to downsample them to these different fractions. The second way I'm going to do is just try to preserve some sort of diversity. So and the second way I'm going to do is we're going to downsample, but we're going to try to preserve the labels that I know these cells have. So if I have a different cell type, I'm going to try to keep the proportion of cell types as in the original data set, but just downsample them so that I have 1%, 10, all the way to 75% of the data, okay? The third idea, I'm going to do the same kind of downsampling, but I'm not going to use the annotated labels from the data. I'm going to use what we call geometric sketching, which is this idea where I can do like effectively clustering, and I'm going to try to preserve around the centroids of each cluster a certain proportion of each cell. Okay. Now on the right-hand side, what I'm going to show you is a result of running a model on these different fractions. So on the x-axis, you're going to see an increasing proportion in data size. On the y-axis, what you're going to see is model performance. And what you expect to see is that if I see more data, that problem performance should go up, right? That's not exactly what you see. So what you see on the left-hand side is, so these are my different schemes, all kind of color-coded together. This is by running GeneFormer. What you can see is a very clear story. This model kind of saturates at 1% of the data. So you might ask, like, okay, how much does this actually replicate across different things? So what we do is we take the study, we're going to do this in many different ways. So I'm going to take these 22 million cells. We're going to do these kind of random downsampling schemes across these different percentages. We're going to do this over many different models and increasing complexity. So models going from just, if I did my model was PCA, all the way down to something more complex like similarity, right? And we're going to do this over different parameter sizes and different inputs, right? We have 4 different tasks that we're going to run. The first are a set of 0-shot tasks, where we're going to think about cell type classification and batch integration on these different data sets. And we're also going to have a set of fine-tuned tasks. One would be cell type classification, and the other being perturbation prediction, where we're going to take Tahoe and split it up into 4 different fractions based on different drugs and different tissues within all these different drugs, right? And in total, we're going to have about 6,400 total experiments across all these different things. This is peer review, basically. So we're going to do this over many different experiments, and then there's some side experiments I'm not going to show. We'll show results for a lot of these. Okay. So the idea of like the saturation idea was really this idea of like, if I take a pre-trained model and I have an evaluation data, can I identify a saturation point? Effectively, what we're going to ask is, at what % of the data do I see that the model is no longer kind of increasing in performance anymore? Like it's saturated, right? So we do this on a bunch of 0-shot classification tasks. So here what you're seeing are our different subsampling schemes, the uniform random case, geometric sketching, cell type reweighted. You're going to see these different data sets on each fraction here across every single different model. And what you can see is that overwhelmingly, a lot of times, models tend to saturate at 1% of the data, and I don't think any more than 25% on this given cell type annotation task. If you want to fine tune, it's like way worse. Like I don't need any data really. I just need 1%, right? And in some cases, I might need 10%, but most cases for a lot of these models, particularly the more complicated ones like G-form and assay similarity, I just need 1% of the data. Perturbation prediction is actually a really interesting one. I kind of want to take a little bit of time to walk through this. So we also did some perturbation prediction. So here you're looking at R squared. So a few things. So this is R squared. The dotted line here is a no change model. It's the model that says, like, what if my perturbation prediction was like, I just predicted like no change from the other state. What you're seeing here are the different colors here. So these different color schemes are like the cell type reweighted, geometric sketching, and then random. This non-pre-trained thing is me just saying, what if we just like randomly initialize the weights and then we just did fine-tuned classification on this randomly initialized model? And that kind of gets you like most of the way in terms of like the ultimate model performance, right? Again, if you want to ask like where does things saturates across all these different things, the answer again is pretty overwhelmingly. It just saturates at once. So we had actually really great reviewers here that kind of pushed us on like these different tasks and these different questions across these different experiments, asking like, well, could it be this? Could it be that? Oh yeah.
Quick question. So it seems like you started out as the smallest data fraction you subsampled as 1%. So did you try subsampling kind of below this and seeing
at 0%? Yeah, like taking like no data, like let's just like randomly initialize. It's like randomly like.
Yeah, I guess for this one, it had comparable performance, but you can imagine that like for 0.1% or like 0.01, right?
We stopped at 1% because every time I did a new fraction, it ballooned in number of things. But that's a good question. Like how low could we have gone? We have a set of experiments in the new sample, which is like not an archive, but I'm happy to send you these results that are on like we do a 0% thing. We include like no data, which was a good suggestion that a reviewer gave us. I'm happy to send you those as like where they kind of compare on those.
Yeah. Cool.
Yeah.
Thanks.
Yeah. Oh, this is exploding. Yeah.
Sorry, just on the last slide for the perturbation prediction task.
Yep.
It says held out cell types. So you evaluated on cells that you like cell types or distribution cells that it hasn't seen. Did you also do it for like seen cell types or?
Not seen. So we try. Yeah. So there's some that are seen. So we do this in like a few different ways. So what we do in Tahoe is Tahoe has a bunch of different small molecules that are trained on across the different cell lines. What we do is that Tahoe's kind of tough because again, place for opportunity. Tahoe's kind of tough because a lot of these cell lines don't actually see any effect across any drugs. What we do is we cherry picked a little bit where we look, their figure 4 shows this E distance between the untreated and treated cells. We took the 4 topmost of those, and then we looked at which cell lines those drugs actually had an effect in. And then what we do is we train on the drug where it had some sort of large effect and held out a random of the other like 4 in that group.
You hold out like an entire cell line?
Oh, well, entire cell line.
So that's even harder then.
Yeah.
So is there a version of the task where you can like hold out a portion of the cells from the cell line?
That's a good question.
I think that's way easier.
Yeah, we didn't do that here. We did the extreme case, again, for this like data explosion kind of question. But I would imagine that you would see these things kind of go up if you were to do it. Yeah. We tricked the hardest of the task of like, if I were an ideal setting, could we mirror that like ideal kind of setting? Yeah.
Here, Lorin.
Yeah.
So they're all basically a flat line around R square of 0.7. I assume that's the similarity between the average training cell type and the held out cell type, right?
Yeah.
So that's, so you don't need a model for that. and when your training cell types are closer to your test cell type, then the line will shift up. And
I'm going to get to some sort of that. Yeah, but you should see it like shift up a little bit. And we're going to get to a little bit. I'm going to go back to training data sets in a little bit, but real quick. Yeah.
Is the no change baseline when you're computing these means, is it using the same test train split?
Same test split, yeah.
So it's only using one, like as you go along, it's using 1% to 100%?
Yeah. Just choosing the same.
Some of the ML field, like people argue that the value of training this foundation model is really to learn this universal embedding and to be able to do some unseen perturbations. So for the baseline being compared, I think one limitation is that they cannot be transferred to the ANSI perturbations. So I wonder, do you have any insight about whether their claim is true or not, whether they can really perform ANSI perturbation wells for the foundation model?
Yeah, my hunch is it's hard. My hunch is like, no. I'll get to like some idea of like where I think we can go for this. Actually, this is a really good segue. I don't know if anyone has like any burning questions, but maybe it's a really good segue of like where I think there are opportunities for this. So we saw this saturation. One thing that a reviewer asked us to do is like, okay, for the models that you can completely mess with their architectures, how much does architecture play a role here? And this is actually quite important for like this question, which is, you know, if I were to take something like SCBI and scale it, so one thing that we do here is we take the number of layers and parameters in SCBI and either cut it in half or scale it up to be double its size and 4 times its size. And these were the baselines that some of these models were struggling to beat before, the HVG baseline, the PCA baseline. One thing that's really interesting, again, this is on cell type annotation, so take this with, you know, we wanted to do something that was 0 shot and not something that was fine-tuned. But you do see this nice kind of scaling thing that kind of happens with a CVI, with the caveat that at some point with model size, our saturation still kind of occurs. With this idea that like, once I go from like 2 to 4, I don't really see any more like real benefit from moving like too large of a scale. So there are a few questions that one could think about going from here. The first is what Davide and just talked about before with the work with Seb, right? The idea is like, maybe if I think about biological priors or biological architectures, that could be one case. So for those who are a little bit new that weren't here before, you know, Davide talked about earlier about this idea of like, if I move out of distribution, at least for the cell type annotation setting, you see this drop off in performance. And one thing that I could think about in terms of recovering that performance is having more of like a biologically driven training objective function, right? And so this idea of moving from just thinking about cross-entropy to something that's more, this like tree-like structure, where I think about this hierarchical cross-entropy might be able to be a solution to improving model performance across all of these different methods, right? And one thing that Davide talked about was this idea, like, you know, if I can see this nice improvement across the cell types, where does that improvement come from? And one thing I want to highlight here is it really kind of comes from this idea of like connecting this across this different tree, right? So one thing that Davide didn't talk about was this idea that like, you know, where you see drop-offs in performance tend to be these places where I have little information about the connectedness across the different cell types. I think someone had asked this earlier about where I see this massive drop-off. A lot of times it happens where I have these isolated points. So there's this one question of like, how do I recover that? Well, there could be some architectural, someone gave a really good suggestion about like a tree-like method when I think about even building these models from the very get-go. But one thing that, at least like when Pete and I looked at this, the one thing that we thought about was this idea of like, whoa, the other thing that really is a cool opportunity here is like the more data we collect so that this tree structure becomes more connected, so I know how different cell types are related to each other, the better these models can also get. There's also like a data-driven question here as well, which is like data can also start to think about improving these different models. One thing that I think is very underappreciated about like ChatGPT is its training corpus. Right, like everyone kind of has a lot of the same architectures now. We all know like what architectures are available. The one thing that's kind of like a secret sauce is this idea of what training data sets go in different models, and that tends to actually be a big separator for a lot of data sets. One thing that people think quite a bit about in language models is you want data in your training corpus that are both related, but also have unique information so that your model can kind of tessellate across like these different types of knowledge. And this idea of how you upweight and downweight certain resources so that a model can then learn and then generalize out of distribution. That's like just a true, like AI is a science. That's a true science that kind of goes into a lot of these methods. So if you take that idea, you can think about, well, what do I need to maybe think about when it comes to training corpuses? And we thought about this last summer with the student, Ajay, who interned with us also from the area, where we thought about, can we leverage like biological knowledge to really think about how to build training corpuses where it's not about the number of cells, the information per cell. So what you want in your data set are cells that have large sources of information per cell, like a bit of information per cell is the measure that we should be thinking about. And so as one example of this to kind of show how we might build really good training corpuses, we took this really big example, the really baby example, where we said, if I took a data set that was trained on just blood cells, and then I took another, and I trained a model on that, and I took another model that was trained on half of these blood cells, but also half of this data set coming from this transcription factor atlas. So the same size of data, but I just split in terms of how much data is represented across these two groups. How well do these models train, or how well do the models perform out of distribution? So let's do a very simple example. If I want to predict, if I take these two models and I say, let's now predict the expression of blood cells, these models both should perform kind of the same, right? So the x-axis, the y-axis you're seeing here is reconstruction correlation. These are squared values. Again, we have the blood model and the blood plus TF model trained on different data sets, but kind of performed exactly the same. But that's because this data is actually in distribution, right? Now I can ask this question, all right, what happens if I train on neurons? So a set of data that this model has not seen on a different cell type that's far out of distribution, how well this model perform? And you get something that kind of looks like this. Now, two things to kind of recognize, the R squared is bad for both models, but you can see that magnitude of difference that I kind of get as almost this like value add that I have from having a set of diverse data, again, very small, but of the same size, versus just having something of all one type, right? And so it started asking us like, what if you were able to now build data sets where I knew I was kind of like information poor, and I started to build data sets that now have extreme value. Again, not thinking about size, but bit of information per cell. So you might ask, like, how well does this kind of finding generalize out of distribution? And it actually generalizes quite well. This idea that, like, whether I trained and tested those data sets on like something that came from the heart or the kidney or again, the set of neurons, having this like data sets that are more diverse, against effectively, these TF atlases tend to express data, cells that have, they just express different types of genes. So we can think about this as like a proxy for more information, right? That value add of having that TF atlas is quite nice. And you can kind of do, this was trained with a linear decoder. So you can actually go back to the model and ask, are there parts of the latent space that I just can't reconstruct with a neuron, in these neurons that I can't just reconstruct with these blood cells? And there's actually a lot of it where the transcription factor atlas kind of has high explainability that is lowly explained by just this blood model. And so you can kind of start to get this notion of like where I should start to think about different things. Now to tie this all back to ex vivo, actually, like this is what the kind of stuff that we're thinking about. So you might ask this question, like, can I start to engineer state heterogeneity for cancer, knowing these value adds that you get from data for AI models? And the answer is that you can. So, what we're doing in ex vivo is doing these kind of like state to function mapping ideas where we're kind of engineering heterogeneity, right? This idea of like, we're taking these like transcription factor libraries across different cell lines, screening across different perturbations, assessing things like fitness. And we're trying to identify both common and tissue-specific cell states and how they map to different perturbations, right? And so we kind of have this like nice different axes, if you will, where you think about as we traverse these different planes of engineered heterogeneity across different hallmark perturbations and how this might scale across different tissues. Now, if you're looking at this, we want to build these AI models that kind of learn across all these different data. If you're looking at this, the ideal case, when we first started thinking about ex vivo, when we first started pitching this, if you are a leader at Microsoft looking to support this or someone, you might say like, do you have to build data that basically for every cell across every plane here, do I have to have a data set for everything here? That seems like a lot of money. Is that the ideal state that we're gonna do? Interestingly enough, that's actually not exactly what we've learned, is that that's not exactly what you need. So one key finding from Ajay's paper was that you can learn parts about one side of this plane, and that'll give you a lot of information about another, right? So here's the full table of all of Ajay's experiments, where he took these different models, trained all on either using like a blood baseline model, and then thought about if I train a model just on blood or just on bone marrow or bone cancer, perturb-seq, and TF atlas, but then I also combined them. So I combined these different data sets, each with blood. How well does it do in testing generating the expression for things coming from neurons, different cancers, bone marrow, and also in blood. And so what you're seeing here is the relative log-fold change of each of these models with respect to this blood model, right? And for a lot of these, you get this really interesting point where, if I train on just blood and then I add other data sets, like they're not actually going to be better than me just doing this distribution. And then we also got some very interesting ones, which are like if I train on blood and I want to test on certain cancers, this is healthy blood, I train on and I test on cancers, I actually do kind of well with just the blood on its own. And then there's some cases where I'm just totally out of distribution, where like the blood model has no shot, and I have to add other like unique data sets in order to improve, right? And so you can take settings from this coupled with the idea that we can actually like engineer representations of models to represent certain cancer models, where the transcription factor idea was this idea like I can take these ORFs and kind of overexpress certain transcription factors to basically engineer cells that express a certain signature of a state that I have desired. And we can do this across many different transcription factors and have this idea of like, how close is a given transcription factor getting me to a certain phenotype of interest? And you can couple that with something that we've done with Ajay's work. So if I go back, this looks like a scary endeavor where we have to do a lot of different experiments, and I don't pipette. And so how do we actually get information from here? But what we learned from Ajay's work is that there are a lot of cases where I might know something about one sort of perturbation in a certain state, and it actually might give me information free about something in another place. And so if we can kind of learn how to traverse this plot in a more computationally nice way, which is stuff that we're doing now, thinking about how to actually quantify where I might be information poor, we can do this in a much more efficient way. And effectively, that's what Ex Vivo's whole thing is about, trying to tie this whole thing up together. We're thinking about how to build these different maps, create new data. And the really important thing that we're doing in our group is thinking about not using all of it, because I don't actually need all of it. It's really triaging the things that just help us the best, train models, learn about them, and interpret them. And that should lead us to our next best experiment. And we can continue this cycle, all with this idea of eventually trying to think about state-based therapeutics. So to wrap up in the last couple of minutes, I'm happy to take all the questions that you have. We have these two different aims. Really what we're doing in the first aim is really how to learn and optimize in silico and engineer corresponding ex vivo models. And the second is really thinking about how to build out these new state of function data sets to learn effective foundation models in the future. So with that, there are a lot of people that are involved. I'm sure I don't have all the names that I should have on here, and I apologize for that. But people have been nice enough to give us money and support, and I'm happy to take any questions that you have. Thanks. Oh, and here are some references in case you want to. Yeah.
Thanks for the great talk. So I had a question about your batch effect correction evaluation with the UMaps that you were showing.
Mm-hmm.
So I was wondering if you've looked at embeddings from different layers, but you could imagine that certain layers learn things about batch, certain layers learn about cell type. So yeah, I'm curious if you've evaluated.
Yeah, we did that with GeneFormer. So on the GeneFormer plot, you will see this thing that has like GeneFormer-6L that looks like that particular layer. We did that with other layers and saw very similar type of results. We didn't do that for all different models, but you're right. There could be questions where certain layers correct for certain things or are better at certain things. That's a really, really, really good caveat.
Some of the questions we had online.
Yeah.
So one question we had was from Stephen Fleming. What do we mean by perturbation effect prediction using SCVI? I was not aware that SCVI did perturbation effect prediction.
Doesn't. It's fine-tuned. So we take the embeddings from SCVI and train a model on top of it. And then, yeah.
All right. Thank you.
Hi, Lorin. Thanks a lot for the wonderful work. And your team shows that the data saturates at 1% for these foundation models, and showing that the gas is enough. But I want to hear your insights on this. It's like, is the car actually designed to take this type of gas pretty well? What if we are pouring this gas into a Tesla while a U-Haul truck or a Ferrari can actually do the work? And because actually the scaling law probably exists, but the current, this transformer-based foundation model was designed poorly, not even optimal to take this type of single cell RNA-seq data, right? Binning, ranking, discretizing the originally sparse data further. So what do you think? It's like if we find another better architecture, maybe we still can scale better. And considering that GPT actually the miracle happens after 3.5, after taking billions of parameters.
Yeah. Okay. I have two like thoughts here. The first is yes. Is a transformer the best architecture? Sorry if anyone loves transformers. I'm not sure if transformers are the best architecture for single cell data. Actually, there's been work on this floor, I think, from people who have thought about alternative architectures that could be better and have shown like really promising performance from that. I think Caroline's group has thought a lot about this question. The second thing that I've thought about a lot is like, are these tasks too simple in the setting of like, are we saturating a lot just because we're not actually taking advantage of, we're not posing questions or tasks that are hard enough. Like there's a task complexity idea here that like with the right complex enough task, you'll hit something that now activates needing a very complex model, and cell type annotation or some of these other questions just aren't that. And so we're like not really aligning tasks with like how complex the models are. And so there's a question of like, we had an intern think about this past summer, which is like, can I start to think about task complexity and should I think about scaling laws with respect to like to that? And I think that might be I think that's a really underexplored area of like, can you align with more biologically difficult things? Yeah.
Thank you so much.
Yeah.
I think that might have addressed some of the other questions we had online recently. So one was from Sam Zimmerman, essentially asking, you know, if these models did better on their tasks, would you see that the models perform better the more data it has? Do you expect to see this an improved data scaling when if you were to have a more complex task? And a related question from Brian Plosky: would it be fair to say that this is another way of confirming that these models don't perform better than the mean, or for a model that is actually better than the mean at perturbation prediction, would you expect the same type of saturation?
Yeah, okay, that's a good question. I don't know about the mean model. The no change model is probably as close to that. Again, I want to be optimistic here and say like maybe that you can see something. There have been interesting Alon's group at Harvard had this paper where they showed two things I think are interesting about scale of data and model scaling with respect to that data. Is Alon's group had a paper that showed that there was a data scaling feature with respect to data quality, and they kind of had some measure of data quality and showed that as data quality went up according to this measure, you did see some sort of scaling law. One underappreciated figure that I thought was in Fabian Theis's group with Till Richer, who interned with me last summer. He thought about, he has this paper called Self-Supervised Learning. And there's an underrated figure to me that I think a reviewer asked them to put in, and it's like a really small panel, but they show like this scaling law with respect to donor diversity that I think is like also kind of an underrated thing. This idea of like moving up in scale and thinking about diversity across donors, whether just like individual cells that you kind of get across this thing might be a really good feature to kind of index on. And so, yeah, I think there are paths forward, but I don't know if we've done enough research to be comprehensive about it. But yeah.
Oh, so I was wondering, I wanted to follow up on the question from the other gentleman. So one explanation also could be that the data is somehow redundant, right? The subsampling. So, but it seemed kind of that your answer to him it was suggestive of that you believe that somehow we can't detect or we can't really reap the benefits of this large data. So can we somehow be sure that the data is not redundant, that just 1% is not containing all the information that there is?
So, okay, yeah, that's a good question. I think there is some level of, okay, this is actually really nice. So
do we need more data?
No, we've been thinking about this a little bit. I've been thinking about this in the multimodal setting with Till. So there have been some really interesting work of people in other areas of machine learning thinking about this idea of which data sets do you get where you get like unique information versus redundant versus synergistic. And I do think there are methods for which you can really start to think about how to quantify that a little bit better. I think what we've been doing thus far has been using like these metadata labels as a way to think about, oh, we've seen enough of this. Do I need to see more of this? And I don't think that's like the right scale to do that with. So I will say that the way that we did the 1% downsampling may not be reflective of this idea of like truly trying to preserve like unique information. It may not be as simple as looking across clusters or looking across cell type labels. So it was just in the way that we did the downsampling. I do think there are other ways to preserve uniqueness, or in some cases, like increasing like the interactions, so the synergy across different data sets to improve model performance. We just didn't think about it in this kind of framework. This is like more like recent work that we've been thinking about. But yeah, I think there are paths forward. We just, in that work, we didn't quantify it in that way. Yeah.
I have another question from the Zoom chat. We have from Amaro. In a setting where most perturbations don't do anything, I think it's not possible to learn a model that does well because most of the gradients are computed from changes in cells that are random or not related to the perturbation. I guess that's more of a comment, but I can also phrase that as a question.
I totally agree. We've also thought about this idea of the difference from the idea that like, a lot of drugs might do something. And so you have like, you kind of discern between this idea that like things change versus things that like meaningfully change. And so, that's another question of like, can we start to generate data sets where you have this sort of meaningful change and kind of detecting that? I think that's more of a biological question that, we are working closely with our ex vivo group to start to try to define. But yeah, a lot of these things end up being noise, which again, I don't think it's a place like Tahoe's fault, right? Like I think if you had to start somewhere, you're like, I'll try to perturb a bunch of different things at scale and kind of see what happens. I think that's just kind of the result of, you don't get a lot of signal from somebody.
Your evaluations seem to be at the level of pseudobulk, but you also look at the scaling with respect to cells. You think maybe that is also a fallacy, given that information you get from single cell is at the distributional level.
Pseudobulk in what way? Sorry.
But you do R squared, which I assume is at a pseudobulk level. Same with cell type annotations, right? These are not considering the whole distribution. Well,
yeah, that's a great question. Yeah, that's a great point. Yeah, I mean, I would argue that I don't know if R squared is the greatest. I think what you really want when you think about like perturbation prediction effect is like a more distributional based like metric. Seb and I talk about this like all the time. Like, yeah, it's just the one that people always think about and people use a lot. But I agree, like this idea of R squared can be conflated or deflated based on like how well you're doing on the cells that change, or if you're over indexing on the cells that don't change. And so you can have a misaligned metric in terms of performance versus like what the model is actually doing across individual cells. Yeah, I think about that. I mean, if you have a thought there, but I know people have been thinking about better metrics to kind of capture the more individual cell type of thing. Yeah.
I have one last question from the Zoom chat. Could a hybrid model from Anthony, could a hybrid model that trains on both genes and expression value and base pairs themselves unlock some of the limitations of expression-centric models, or are these two data types just too different to be useful in pre-training?
I mean, maybe. I think that's a really good point. I mean, like, I get this question a lot, which is like, do we, like at ex vivo, think RNA is everything or the end all be all? Like, I think, no, I think multimodal things are the way that everyone's kind of moving. The real big question is how you start to integrate across modalities, right? And like one strategy people have had is I can just take one model trained on one thing and then train a model on top of that. I think UCE sort of leverages some of this kind of strategy. I don't know if that's the way to kind of go, because you kind of lose, what you really want to do is capture the interaction across modalities, which I think is like the real challenge. Like, how do you do that? But I do agree. Like, I think incorporating other modes is to get a more holistic view of what you think cell state might represent is a good path forward.